credit normally include practical classes. So 3 credit normally do not have practical classes, right? And this is a prerequisite, this particular course is a prerequisite for second year, which is medical microbiology. So when you go to second year, if you pass this course, you go to the medical microbiology in second year. Talk about lecture. Lecture every Tuesday. I think you'll find where is it very good. And from 4 to 6, we will have all physical classes for the lecture. We do not have any online or live lecture. For tutorial, we supposed to have 5. When you do your course selections, you're able to choose one out of these 5. But this so far, when I check, the number of students in this particular day is only 9. So it's a very small number. So I hope that this group of students, which I think I'm going to announce it, can actually move to other tutorial classes. Right, so these are the things. You need to know which group it belongs to, right, according to my teaching plan. For practical classes, I think some of you have read my teaching plan that I think I've removed one of this, but unfortunately, I think there's quite a number of students registered to the one that I want to remove. So I remain it, I cannot remove it. So we remain as 4 practical classes. So you also do it according to your group number to attend your practical classes. All of them will be conducted in the cell biology lab at level 4 at the university building. Any questions? No? Alright. What is it? Course learning outcome. I think all of you will be familiar with the microbiology, which is very important as any medical science program. Microbiology is a very classic module, but it is very important because we have microbes that living around us. So we want to know them, we want to understand them, we want to actually protect ourselves from those who are pathogenic, and also we want to utilize them. So your course learning outcome will be to apply the property. You need to know the property of all the microorganisms and the impact of them that relate to your human life. So not only they are pathogenic, but daily food that we eat, they may come from this microbe, right? And the air we breathe is also contain the microbe. So we need to know their relationship with human life. Secondly, we need to also know the impact they can do on our life. I think from the last several decades, maybe like 50 years ago, people just trying to understand what is microbe, what is the impact of microbe that do on our health. But now we are not really focused on that only, but we also need to utilize what kind of applications, what kind of microbe that we can use to solve our problems. For example, we want to have the microbe that can degrade the plastic, right? Degradable plastic that we can eat through of the plastic or oil, and also to actually use it as a technique, a tool to do a lot of molecular work, a recombinant work for therapeutic, and also for a lot of drug therapy. So this is what they can do. We will go, I think we will go one of the chapters should cover this too. And finally, and also very important, if handling microorganism is different from handling others, like your biochemistry, your biotech subject. So handling micro, the first thing we need to be conscious that we do not want to have contaminations. So that is a particular set of technique you should learn from handling the microorganism. So this practical class is very important. You may not like it. I think some of you, at least half of you, may not. So I don't really like the study about microorganism because I've been studying since my high school. But at different level, and you will have a different set of rules that you need to take precautions and different technique you have to learn when you come to deal with the microbiology. So 1, 2, 3. So now we talk something very practical. Assessment. Similar like others, your final 60%, lab report 20%, individual assignment is 20%. So altogether is 100%. They are individual lab report and individual assignment. So there is no group assignment. Good. Some of you might think that is good because it is very difficult for you to find another group member. You cannot actually form a group. My purpose is not allow you to form a group. It's just that the setting of the assignment is easier to be an individual assignment rather than a group assignment. Right? But you will practice how to actually work as a team in other subjects as well. There will be a group assignment in other subjects. To get the detail of practical report assignment, so it will be a research article and the MCQ. And this assignment has part A and part B. Each part of it has one short article, one long article. The short article, quite easy. It's like a reading a newspaper or a magazine. So it's all in layman terms. So it's easy to get to know. The long article will be a research article, but it will be in a review format. So it's quite long. I think up to 10 pages. And once you read it, you need to answer the MCQ. And each part have 10 MCQ. And you're going to do it on e-learn. Very similar to the format of cell biology. And you do it on e-learn, and you will have 3 times to try on it. I've given you a period, I think I will have the next one. I've given you a window of 2 weeks. So within 2 weeks, I release the article, and then you go in your e-learn. You can download the article whatsoever, and then you go to try to answer the MCQ in 2 weeks. But within 2 weeks, you can only try 3 times. So you need to find a time really that you have an hour or so to do it. Don't go in on and off, right? So that assignment will be released on 17 June. So within these 2 weeks, you have to get it done. And each of the MCQ is 1 mark. And the result will be released on 15 July. Okay? Any questions? No? Good. Practical class. We have 5 practical class. So these are the 1 to 5. You have the timetable already on your teaching plan. So, what are you going to do with the practical class? You need to follow the rule, must have a lab coat. I think quite many of you do not have a lab coat. Try to get one for yourself because you're going to wear it for the next 3 years. If you have the lab coat from maybe Sunday College, maybe from another college, it's fine. We don't really care about what is the logo putting on your lab coat. As long as there's a lab coat, it's fine. Okay? of course, for those who have yet to buy a lab coat, I think the lab technicians will prepare the lab coat that ready, just in case you forget to bring a lab coat. So we have the lab coat in the lab, but I don't think that you want to wear the lab coat that somebody else has worn before. Right, so bring a lab menu. I already uploaded the lab menu on eLearn, so you can either download it as a soft copy or hard copy, whatever, but read it before you come to the lab. Is because if you're not, I think that there's quite a lot of steps that you need to follow. If you do not read it beforehand, I think when I explain it, normally I will give a briefing before the class, before the practical class. If you don't read it, you may not understand what I'm saying, and then you start to reading all the step by step, and then you delay. So it's quite packed. I think cell biology, in a way that I think finish that within an hour or so. But microbiology, I think is quite long. Sometimes it's more than 2 hours. Believe me or not. It depends on your individual. When you look at the microscope, some of you very easy to find the answer. Some of you even taking like 20 minutes, couldn't get the answer and keep going. So it could take quite long. And we provide the marker because you need to do the labeling on plate, on tube. Very important to make labeling. And if you do not want to use the one that provided by the lab technicians, you can actually prepare your own. Okay, make sure it's waterproof. And no open-toed shoes in the lab. This is standard. Okay. Most of you, right? And also cut your hair if you have a long hair. Do not let your hair like sitting down like this. Just cut your hair, right? In the lab. Alright, come with the assessment. Assessment, I think I make it in a way, easy way. So you will have to submit 5 lab reports because we have 5 practical class. 1 practical class, 1 lab report. It's an individual lab report. I already prepared the template for you. So you just need to use the template to fill in and to answer the questions. Similar like cell biology, what we do is that, forget about this one first. You have the template, you download from eLearn, and once you have done, you finish, and then you submit your lab report in the next practical class. You bring a hard copy, a printed copy. This is something I ask for your favorite to do for me, because if you want me to mark, can, no problems, but 109, it would take like ages for me to mark individual report, and you may not get the feedback. I found this is the best way. We come back the next class, we submit the lab report in the next class, next practical class, and we mark it together. Same times, I can give you feedback. So you get the feedback at the same time. You doesn't need to wait for a long time to get the mark. Now, this is something different. For microbiology, you need to grow the bacteria or whatever, the bacteria you need to grow. Normally, it takes like overnight, 24 hours at least. Now, you do it, the practical class today, you may not see the result. You will see the result the next day. Normally, I will ask you to come back the next day to view whatever on the agar. Right, clear? So this is something that you need to do extra, come back, same times. Like, I think I opened the lab for the whole day, you come to view your result. And this is what this means. Normally it's the next day, unless the next day is a public holiday. Any questions for your practical class? No? final exam. And sections A, B, 30% for section A, 70% for section B. MCQ, 30 of them, 1 mark each. And for 70%, all of them, your 6 short questions, all of them will 70%. Right, so it's not, doesn't mean that each of them like 12 marks or something. Some of them could be 8 marks, some of them could be 12 or the 14. Depends on the questions. And the difference from your cell biology is that this is 2 hours paper. Your cell biology is 3 hours. So you have to get it done within 2 hours. Should be okay, I think. Since it has been done for many years, I'm not the one who started with that. I do not know why they set it as 2 hours instead of 3 hours. And I think because the cell biology have an essay question, that's why it's like got additional 1 hour.
No essay.
Any questions?
Oh, this one. A, B only. Oh. That's why I got 10 more. MCQ.
10 pages of slide.
Check-in code for that?
This for the assignment. Not for this month.
For the revision? iZone? I mean your attendance? No, the attendance just now. It's okay, we'll come to me after the class. For textbook, I think there is an update on the textbook. I don't know. Some of you think that you should have a textbook. Go ahead and get a textbook, which are quite popular. I think this the first compulsory textbook is by Joanne Wiley, which is the Prescott Microbiology. Currently, they have editions, 12 editions, right?
Your one is this one.
And the other one, whether you have it or not, is okay. This is by Totoro, which is the microbiology and introductions, which has the 13 editions from Pearson's. So any others, basically you can actually Google online, and there is a lot of free textbook available, and you can use it as well. Not necessary that you must have this. I also not too sure whether the library have it or not. And this, I think this is covered. Now, I have teaching plan. Teaching plan is very important. And I have students come to me before. She missed my practical class, and she missed one, and that is the last one. And therefore, she is not able to attend anymore. And she tell me that she come to the class based on my iZone, based on the iZone that indicate where is the class, when or where. However, do not follow. You follow your iZone because iZone is a general, giving you the date and your group number. But if you want to attend, because particular classes, you need to follow my teaching plan. Right? So I think some of you already used to my teaching plan. I have the date and written and the time written on my teaching plan. For example, this is the lecture. So for the lecture, I think we do not have any public holiday on Tuesday. So every single week, we will have the lecture, which is very good. So that we will have the last 2 weeks, we'll have no lecture. So remember, every single week from now on, you will have a lecture. So I will written in this way, all right? Now, what is this? This is a practical class. So you will see something like that. I will not put anything for the first 2 weeks, because the first week, as long as I hit one of your practical class having a public holiday, I will cancel all the practical class on the same week, because everyone is doing in the same pace. And therefore, week 2 has no, because of the Labor Day, I don't know which day. And so normally first 2 weeks, and I think from the new student, normally, unless the lecturer specifically tell you that will be a practical class on week 1, Normally, there is no practical class in week 1 or 2. 3, we have the first practical class. I should have 4 groups here. Sorry, I will change it. And then lab is not available. The reason is that we do not have enough lab. So I'm sharing the cell biology lab with another lecturer. So when her practical class is on, I have to be off. So that's why that is not available for me. It will be available for another lecturer. So that is a visa day, no week. You have to follow this week. This is week 1, 2, 3, 4, 5, 6. All right? And accordingly. Clear? Good. So tutorials, same thing. You follow my teaching plan as is written, the date here. So this is a date you should attend your tutorial. Something like that. Now, oh, should I go back? I forgot to give you. Yeah. For your practical class, I think I make a remark here. Attend all your practical sessions. Why that's important? Because only when you attend your practical class, then you're allowed to submit your lab report. Imagine that without attending the class, how do you know what is the result, what experiment you have done? So you're not be able to write your lab report. If you are not be able to write your lab report, you have no mark for your lab report. I have students not coming to the class and then ask me whether I can submit the lab report. Then I say, how do you get your result? Then you say that, oh, I get it from my friend. And then getting from a friend is not a good idea, it's a plagiarism because you're getting somebody else's result. Yeah, put it as your own result and put it in your lab report. This is not encouraged, and you didn't actually hands-on and do it. How do you know what does it mean in your result? This is ethically not allowed. So please attend your practical class. Even only, I think each of that, if you divide it, is 20% for your practical class. If you divide it by 5, 20 divided by 5, for each of them 4 marks, right? 4 marks is your final mark. If you have these 4 marks, even that you get 3 marks out of 4, it really helped in your final mark. Really, really helped you in your final mark. I have students not attending the class, not submitting the lab report, even asked her to submit, not, and then, so it's got 0 for 1 or 2 of the practical class. What happened is that she failed, and she failed, the reason is that she failed, of course, failed the final, and basically your coursework assessment can help you to pass. However, since not attending, not submitting lab report, 0 mark, and finally failed the entire course. So it will impact very, very much. Unless you are very confident, no problems. I can do it, even I skip my class. So I advise you, do attend all your practical class and submit your lab report. Can you change your practical class, swap here and there? Yes or no? My old student? Everyone laughing.
Can.
Because they have done many times. I allow you to do that, but don't do it and take it as you like. So the reason that I'm not encouraged, you can do it. For example, I have, you're very sick, right? You're very sick, you can't even get up from the bed. So you cannot attend. You just wrote to me and tell me which other, you know all these practical classes. You told which one that you can attend. But the only certain circumstances that you can do it. I allow you to do it, but don't take it as an advantage that just change as you like. Because all your things, the material prepared for your practical class is prepared by the technician. So she knows the number of attendance. If you suddenly there are extra 6 students joining the practical class, then she need to prepare from scratch. It's very problematic and make inconvenience for her. So remember, don't change it unless you think that you really cannot attend. It's okay. I prefer that you change to other practical class, at least attending one of them rather than not attend at all. Okay? For tutorial, I think my old student can say that you come and go as you like. Huh? Just tell me your attendance. As long as the capacity of the classroom can fit all of you inside the classroom. I don't mind if you miss, you just try to skip whatever, but at least attend one of this. When you attended, don't forget to give me your name and you want to take your attendance. Okay, and all right. So if you want to talk to me, I don't want 109 of you talk to me every day. But if you really have a questions and you want to ask me something, you can write to me. And please make an appointment if you want to see me. And I'll be very busy doing tutorial and practical class during the week. So just remember, this is my email address. Write me and make appointment. And yeah. All right, any questions? Oh, I forgot to talk about the tutorial. That is one group. Group 2. I wish for one of these, I think group 2 is on Tuesday 12 to 1 p.m. Right? And this group is only have 9 students enrolled. So I'm wondering this 9 students be able to change to other group. Ellen? Chan Ji Ming. Are you here? Okay. And Chong Yu Shen. Good. Go Moon Hao. Kazi. Moon Jalin. Tashsin. Oh, okay. Li Kai Jia. Right. Lo Yon Sham. I may not pronounce it properly. Lo Yon Shen. There. Okay, sorry, I couldn't see you. Lok Hui Ping. Ryan. Nhan Wai Kong. Anyone? Ryan? Oh, sorry, Ryan. And Trisha. And Gomez. Trisha? Okay. So, would you be able to switch to other group? Okay? Do me a favor, because I cannot have 2 hours plus another 2 hours on Tuesday. That will be total 4 hours. I'm going to stretch my voice very much. So, these 9 of you wrote me an email. Tell me which group you want to change to. I'll be able to actually do it for you. Alright, we do it in this week or next week. If it's possible, then we quickly do it in this week. Alright, thank you. Any other questions? Hybrid? No. The feedback that I got last semester, that student highly request, I think not all of you are quite okay, doesn't really matter. I think 1 or 2 strongly suggest to have a physical class rather than online. I do not know why, because they say it's less interactive with other students, don't see each other very much. I don't know why they want to see each other very often. But this is the reason, and they say that the online is actually, they couldn't pay much attention. So they prefer to have physical. So therefore, this semester, I totally changed it to a physical class. Yeah, unfortunately, there is no record, and those who are absent may not be able to go back to see their video recording. You set it up for me that I can do this. I do not know how they set it. Possible, at one time, recording difficult, live possible. I think some of the seminar have done it live. For example, people outside of campus, they actually see the people talking, they see the live streaming. But recording, I don't think so. I haven't seen that. Yeah. All right, any questions? No.
You ask me, you are.
Don't know. I'm going to see. What is it? All right, we're going to have the very first one. Don't worry about this one. This will not be in your final exam, just introductions for microbiology for today only. All right, very short. The only thing is that I need the keyboard to click the next one. Right, this is particularly is an introduction to tell you what are the microorganisms that you need to learn from this course. Microorganisms not only include bacteria, but there are other, not viruses, not bacteria, but there are others. So to know about them, what are they? And also you'll be able to differentiate and these microorganisms. All right, more or less throughout this course, most of the time you learn about bacterial, archaebacterial, protists, not really. Fungi, yes, I think quite important. Virus, yes, at least there's one chapter focused on virus. So those are the so-called microorganisms that are covered in this course, but somehow that they are different and you need to know about them. So what is the definitions of microorganism? Microorganism is microscopic because they are very small. You need a microscope to see them, therefore they are microorganism. And what about microbe? Microbe showing here is relative, is a new term. So microorganism, normally we refer from the many decades, we already call all these microscopic organism as microorganism. Microbe, I think, is for the last 2-3 decades, they start to have this calling them microbe, but basically they are the same thing, right? And so they have include bacterials, archaebacterial, fungi, protozoa, and algae. And they can be single cells, meaning they have single cells, or they can be multiple cells, meaning multicellular organism. And one important, if you want to actually classify the microorganism from eukaryote, or some of them eukaryote, or from animal or plant, let's say that. They are very simple, they do not have differentiated cells. What do you mean by differentiated cells? So all the bacteria, even that you put them together in a culture, in population, millions of them, they all are identical. Differentiated cells meaning that you have a population of cells, but they are not identical morphologically and functionally. For example, brain cells, our nerve cells, epithelial cells, right? The cells from kidney, liver, they all are different, so they are differentiated cells. But for microorganisms, they are very simple. they do not have highly specialized cells. And that's what it means. And of course, they do not even have the distinct tissues. Only animal or plant or higher, what else? Protists? No, I don't think so. They don't have it. Right? So they do not have higher organizations of tissues. Now, the thing is that virus, I think most people now agree that viruses are not a living organism because they do not have their cellular, the cell membrane. They have to actually live in a whole cells. So therefore, they don't consider it as a living organism. However, since they are small, they have been so long have been classified as one of the microorganisms. Therefore, we will also include the virus in one of the chapters. But most of the researcher or scientists now agree that viruses are not living organisms because they are not free living. They cannot live by their own. And prion is a protein. I think I will actually have a slide to talk about them later. Now, interesting. How about 5 kingdoms? And you know all these living organisms, but you might have to classify them so that easy when we talk about them. Right? Some of them is very clear. These 5 kingdoms. 5 kingdoms actually is proposed. If you Google it, there will be 5, maybe 3 kingdoms, 5 kingdoms, 8 kingdoms. It depends. But the most popular and widely accepted is the 5 kingdoms. It is actually proposed by an ecologist. His name is Robert Whittaker. That he proposed it in 1969. There are 5 kingdoms, and these are the 5 kingdoms that he proposed. And he proposed it based on what? Based on the cell structure, whether it's eukaryote, single cells, or even multiple cells, but they are identical, or eukaryote, have multicellular, or mode of nutrition, whether those organism is autotroph or heterotroph, meaning that they can synthesize the food by themselves, or synthesize the energy by themselves, or they need to take the energy from others. Number 3, he defined the classifications based on the body organizations, meaning that when you look at it, whether unicellular or multiple cellular. Okay, very simple. And the one that, remember, all these 5 kingdoms that he defined, which is kingdom Protista, and Monera, fungi, plantae, and animalia. So 5 of them. And I forget about the plant and the animal because they are too big. I'm not going to talk about it. So for microorganism that you found and you're going to cover in this course, you found them in the kingdom of Protista and the Monera and also the fungi. These 3, 1, 2, 3, they contain the microorganism. All right. This Lynn Margulis, very famous. I don't think you know her. She's famous at my time because I'm coming from a background that I come across with her. She's famous. She has published this book, 5 Kingdoms. It's expansions from Robert Whittaker. So he further expand and modify and define even clearer all the characteristics of the organism. And she's famous because she's talked about the endosymbiosis. You learn about endosymbiosis? An organism living in another organism. You learn about mitochondria. Mitochondria originally now is widely accepted. Or mitochondria is basically coming from bacteria living in another bacteria. And then they actually become mitochondria. This is what she proposed. Chloroplast and mitochondria originated from a living organism. But at her time, I don't know what is her time, very old. She actually passed away already. No one accepted. She submitted her paper regarding endosymbiosis. I think at least more than 10 journals, and no one accepted her paper because they don't think this is possible. An organism living in another organism. Until now, of course, we have a lot of sequencing. We have the DNA data. We can actually sequence the genome of all the organisms. We found the relationship. Mitochondria actually has a genome closely related to cyanobacteria, let's say. So this is now we widely accepted, but at her time, unfortunately, no one accept what her theory. So what she also make this kind of that. So from Monera basically is bacteria, archaebacterial. What difference that you make between prokaryote and eukaryote? Anyone? So first of all, organism she divide into two. So what are the difference, the big difference between these two? Yes, you have the nucleus. Anyone else? Sorry. Cannot hear. Still cannot hear. What is that? Organization of genetic informations. Organization of genetic, what does that mean? Is how you organize in a nucleus. Smallest, okay. Yes, the main difference here, first thing is that eukaryote have a nucleus, has a nuclear membrane to cover the genome, but prokaryote no. That's the thing. Alright, so very easy. And then from eukaryotic cells, you have unicellular and multiple cellular. This is also easy. What make you that protista put it in the unicellular? In fact, most of them, the majority, I think 90%, they are unicellular. That's why they are not considered not multicellular organism. And here, you actually define this group from this group, and because of what is the difference between these two groups, with cell wall and without the cell wall. Now, they have, that's very easy, right? The animals, we do not have cell wall, but plant, they have cell wall. What about these two? Plantae and fungi. Why? Fungi actually, long ago, they actually include in a kingdom of plant. However, they basically take it out and they has its own kingdom. Because they found very different. First of all, this is phototrophic and this is heterotrophic. Phototrophic, they're using the chloroplast to get the energy from the sunlight. But fungi, remember, cannot do that. They cannot synthesize any food or energy from the sunlight. They are considered as a decomposed, that they actually to degrade, using enzyme to degrade any food, whatever the decays compound from around in the environments. So they are decomposer. Therefore, it's very clear that she actually can distinguish all of them into a subgroup. And again, emphasize that microorganisms can be found in Monera, Protista, and also fungi. Now, go one by one. The first one is the kingdom of Monera, and it's a prokaryote, only two. Okay, two living organisms. You remember here is one is bacterial, or you call eubacterial, which is a true bacterial, and the other one is archae or archaebacterial. And what are the difference between these two? They are similar. They have a single cells, and they also have cell wall, no nuclear membrane, and they are also very small, the cell size. So what make people actually put them into two different groups? Okay, good. One living in a very extreme environments, right? And the other not. So which one that living in the extreme environment? So is, first of all, if you look at the habitat, that the archaea always living in the extreme environment, salt lake, also the volcano, hot spring, high temperature, So they are heterophile. And however, the bacteria, always remember the bacteria living around human. So the most comfortable temperature for most of the bacteria will be 0 to 50 degrees Celsius, more or less like that. Whatever we can stay in the temperature, in that temperature, they can endure that temperature. Of course, some of the bacteria is quite extreme. It's like they can similar like archaebacteria. Some bacteria can stay in a very high temperature, but the majority of bacteria stay around human. Cell wall, they are different in cell wall. Peptidoglycan, we will talk more about that. Peptidoglycan is the name of the cell wall that describe the cell wall bacteria. Archaebacteria, they do not have peptidoglycan, but they have something similar called pseudomurin. Yeah, similar to the peptidoglycan from the bacteria. But instead, they have something like the S layer, which is the paracrystalline protein surface layer. This one here. So this is the transmissions TEM, transmission electron microgram, which you see the layer here. This is pseudomurin, quite similar to, but it's not the true peptidoglycan. But if you see the S layer, which is very, very different. Basically, they are one or two type of protein that they align, organize it, align in a very well that they can, like a crystal. So this is an S layer outside to protect the archaebacterium. And in terms of the membrane, underneath it will be the membrane, right? The cellular membrane. And the fatty acid is quite different in the way that the glycerol, this is a fatty acid. So active fatty acid has an axial change. And the one that link this to the chain, that is a glycerol here. So either the linkage for, cannot see, for the archaebacterial, it will be an ether linkage. And for the bacterial or for human, including human, is we have the ester linkage. Ester linkage is contain 2 carbon and 2 oxygens. However, for the ether linkage, it only contain 2 carbon and 1 oxygen. So there's a difference between the ether or ester linkage. So the lipid compositions could be quite different. Intron. What is intron? Coding or non-coding? Non-coding. So intron is non-coding. That one is about coding one. Exon, good. Remember, no coding, no intron in bacteria or Monera. Intron only present in the higher organism. Even in eukaryote, for example, yeast, they have most of the gene, they do not have intron. The higher, the more advanced of the organism, they have more intron. Intron actually is something, is not extra. People think this is extra because it's not coding. It will not be made into protein. I mean, the coding sequence will not be actually translate into protein. But in fact, this is very important, all right? Unfortunately, I do not have a chapter talk about the genetic or genomic of the microorganism. I'm not sure in your medical micro you will cover that or not. I would think that's rather important to have something talking about genetic of the bacteria. Yeah. Sporations, which is able to make a spore. So bacteria can make spore, but archaebacteria cannot make spore. Why do they make spore? Is a way that they survive in the tough environment, right? So only certain bacteria they can make spore. Allow them to make spore to get over the very tough period of times. And then they germinate and become the vegetative cells. So yes or no? And then the pathogenicity, very simple. A lot of bacteria, they are pathogenic, which is very harmful to human. And for the archaebacteria, they are non-pathogenic. Why? Because the archaebacteria cannot stay in our body. So this is not the right temperature for them. They are normally stay in a very extreme temperature and very extreme environment. So our body is not ideally are not good for them. That's why they are not harmful for human. All right. Now we come to another kingdom, fungi. Fungi can be divided into true fungi or fungus-like. Forget about fungus-like, and there are a lot, and they are not very clear. But classically, we can actually dividing the true fungi into 4. They are I think I have a spelling mistake. Phycomycete. Phycomycete, this is a spelling mistake, I changed it. Ascomycete and Pasteomycete and Deuteromycete. Right? Phycomycete that have 2 very typical. One is the bread mold, Rhizopus. So something that you see they grow on the bread, which is when you have a close look at it, it has a lot of fiber-like structure, and this is the fungus called Rhizopus, and they are the bread mold. And also some of that tomato, they also have so-called muco. They actually is another kind of fungus that's grow on the plant. So typical, there are 2. For the Ascomycete and the Pasteomycete, they are the very large group and the main group of the kingdom of fungi. So for the Ascomycete, they include yeast. Remember yeast, mold, and this one is Perziza. This is one, you familiar with that? This is the food that we always eat in the Chinese dishes. No? I think some of you definitely know that we commonly take it as a food. Then they are part of the, they are the fungi. We call Perziza. These are the typical the fungus, the fungi that we see. So yeast is single cellular, but the mold and the Perziza, they are multicellular. So within Ascomycete, they have single or multiple cellular organism. Pasteomycete, they cover most of the mushroom. So mushroom also belongs to the fungi. Alright? And this Deuteromycete is the fungi imperfect. They call it fungi imperfect because there are still a lot of information from this group. They still unknown. For example, their sexual reproduction cycle. They are having problem to culture them in the lab. Therefore, they not be able to study. So therefore, I just leave it blank because they have a lot of information they still not. Therefore, they call it fungi imperfect. So you have some idea, just give you some idea what is the kingdom of fungi. You have yeast that you use normally to make the bread or to actually make beer. You have the mold, which normally grow on your fruit. And you have something similar like mushroom, like this. Of course, you have the mold that grow on the bread or some tomato. Now, just some characteristic of the fungi. Yeast is unicellular, as I mentioned. Mold and mushroom is multicellular. And there is a characteristic very clear for the mold and mushroom. They form a so-called hyphae. What is hyphae? Hyphae is something like that. So mushroom, as you see, as a intact, right, body. But actually, under microscope, you will see there is a lot of fiber, right? They actually cross-link to each other as a network. So these are the hyphae. Each of them is one piece of hyphae. And of course, here this is another one. And heterotrophic, what does that mean? As I mentioned, they are not autotrophic. Autotrophic meaning they can synthesize the food by themselves using the energy from the sunlight whatsoever. but they cannot. They actually has to absorb from any decaying compound in the soil, so they can only decompose anything the organic to get the carbon and energy. And they also associate the plant root, which is a good fungus. For example, mycorrhiza, they actually grow at the root of the plant. So you will see this one in comparison. This is the one with the fungus, which is grow better. This one without the fungus is grow shorter, sort of. So actually it's mutually benefited each other. So yeah, this is the mycorrhiza. Alright, so this is a very scanning electron microscopic, and they put on the color, and you see this is the yeast cells, very typical yeast cells. And sometimes you will see a bud coming. I think like something like that, that's a bud coming. And this is the bud, sorry, this is the bud coming out of the cells. They have through the budding for asexual reproductions. Alright. Now, for the very last, I think this is the last, the protista. Protista, I think this is something that we have least familiar. The reason is because they are not really pathogenic to humans. Some of them, yes, but majority, no. Because majority, they are living in the ocean. And ocean, which is far, far away, because we are on land, but they are in oceans. And they are very diverse. It looked like for these 5 kingdoms, anything they cannot group in any other kingdom, they put into this kingdom of Protista. So therefore, they are very, very diverse. They are mainly unicellular, but some of them, it can be colonized in a chain form or in any kind of form. Alright, they are much larger. They are also very, very much, we are not well studied and very much unknown. And they contain many of them, because as I say that anything they cannot group in any other kingdom, they put here. So they have the algae, dinoflagellate, slime mold, water mold. And they look very simple. They look like this. This is a dinoflagellate, this is an algae. But genomically, they are very complex. They even have, you know, the human genome size is 3.3 gigabase, but they can have very small. I think it's, I don't know, it's maybe like 1,000-fold less, smaller than human, but they have 1 gigabase as the genome size. And not surprised, some of these organisms even have the bigger genome size than human. And therefore, they are the least studied because they have big genome size. Normally, people won't spend money to study something that's interesting, but may not be very useful to human. So that's why they have the least study, all right? But they are also very interesting, this type of animals. Not animals, organism. And they're very pretty, very, very pretty. So if you look at this is their structure, right? One of them, the scanning electron microgram, actually produce this guy. This is actually their structure. It's a very, very detailed, precise structure that can create it. And they also can classify them into the plant-like, animal-like, and fungus-like. So the plant-like, of course, is the algae. Those that algae can undergoing photosynthesis, they call it plant-like. Protozoa is the one that they actually cannot synthesize the energy by themselves. They has also seen as fungi. They need to actually take the nutrient from others. So what do? They are more tough, they can move, and they eat others, organisms smaller than them. Anything smaller than them, they eat. So for example, this didinium, they inject the paramecium. You know the paramecium is a very long one. This is a didinium, is the same size. You see how it's actually engulfed and then take it into the cells. So they can do that. So this is a protozoa. That's why they call it animalite. And the last one is slime mold. And they also have two kinds of life cycle, plasmodium and cellular slime mold. We will have, I think, a few slides later in other chapter to talk about that. Water mold is basically not also well known, but they are not important for human, but they are causing the disease in plant. Slime mold, because of one of the organism called Dictyostelium, I think they call it Dictyostelium here. This organism, we call Dictyostelium, very well studied. Do not know why. Some of the researchers picked it up and studied it very well, and therefore we know a lot about the slime mold, and it become one of the model organism. Yeah, one of the model organism, and therefore we know a lot about the slime mold as well. Now, this is the slide you try to, because slime mold is more like a fungus-like protist. And there are something that different between these two. So slime mold, of course, they belong to a different kingdom, first of all. And then the cell wall, chitin, the fungus, they all have the chitin as a cell wall. But in terms of the this slime mold, there is a cellulose. Right? They have a hyphae. As a hyphae is a thread-like structure, is present in the fungus but not in the slime mold. Mycelium, when hyphae accumulate a lot, it becomes the mycelium. Of course, if you do not have hyphae in slime mold, you do not have mycelium in slime mold. Like the last one, that they present in the plasmodium structure, meaning that in the cells, in the cytoplasm, normally they have in their life cycle, in one stage of the life cycle, the slime mold actually multiplying the nuclei into many. So in a single cells, a big cytoplasm, there is multiple nuclei. So it's multi-nucleated structure. But normally what we think is that as long as you have duplicate your genome, you might have the cell division, right? To separate the nuclei. But in the plasmodium structure, the nuclei been duplicated, but the cells are not divided. And therefore they call a plasmodium structure. It's present in slime mold, but for most of the organisms, including the fungi, they do not have that. All right. Second last slide, probably. Okay, this, just to let you know how amazing of the protozoa or dinoflagella. The dinoflagella called Alexandrium. You see when there's not only the Alexandrium, there are many more. They call so-called the red tide in the ocean. When you see something very colorful, something in the seashore, you will see that they are actually full of nutrition because they have a lot of dinoflagella actually here. Suddenly there is a bloom of the dinoflagella. So dinoflagella, they have the color. Alexandrium has a pigment of red color. That's why it's showing infrared. So it's actually nutritionally rich in this. But of course, it's also same time is a problem, the red tide. Why? For the fisherman, is a problem. Why is it? Anyone come across? The fish? They will eat the algae. What the fish eat the algae? Not really. Not really. There are more prominent than that. All these protozoa or dinoflagella, they also secrete toxin. So when they accumulate, when they are not in a small amount, that's fine, because ocean is big. But with that, huge amount of that, they also produce releasing toxin. So the toxin, same time, killing a lot of fishes. So you will see a lot of jellyfish suddenly, they all die, right, on the seashore. So this is a problem of red tide. It's something good that you have this meaning that is nutritionally rich, a lot of organisms living, but same time they also produce toxin. So it's not something not good. The other one is very amazing. You see this before? I seen this before. I go to one of the port and I collected the seawater. I mean, when there is a high tide, I go to the port using a bottle to collect the seawater, and then I just shake the bottle. I see something lightning. It's a bioluminescent, blue color. Very light, it's lightning. And they are actually done by this called Nautiluca scintillans. This one. They have a pigment. So the pigment can emit the bioluminescence. So when you go to this, you see sometimes there is the light on in the seawater, and they are caused by this kind of the nanoflagellae. Have you seen before? Yes, right? No. You must find a time to see this. The nature is amazing, especially that's something that unknown in the seawater a lot. Okay, the cellular infectious agent, which is, we call it any other than the five kingdom we talk about, and those are the infectious agent, meaning they cause humans sick, and they are of course, the virus is one of them. So this is the structure of bacteriophage. This is the virus. They are quite different in the structure. We will have one chapter cover that. So they cannot survive outside the host. And they are very simple. They only have the RNA, either RNA, DNA, and it will be encapsulated by protein. And viroid, we also classify them as a cellular infectious agents because viroid can cause a disease in plant. For example, this called coconut gadang viroid. Yeah, they cause the coconut tree very sick. They got infectious. And this viroid, we don't call it virus because they simply has a very simple form. They exist as a RNA, single-stranded RNA like this, folded into a secondary structure. And this is viroid, not even with the capsule. So they are simply like this. We have prion that we actually quite know because of the mad cow disease. Or we have this protein. This prion is a protein. Somehow this protein can, when they have been mutated, they can influence the protein next to it to change, become a mutated form. So this can, when you're actually accumulating enough prion in your brain, you're actually damaging the brain, and is somehow, we have a common name called mad cow disease, or it's called the fatal familiar insomnia in human. So this is the one that is a cellular infectious disease. A cellular, meaning that without the cell membrane. Very last one. So as we have made a conclusions, why we want to study bacteria? Because all these microorganisms, because they're living around us. They're living in the soil, Antarctica or Arctic oceans, in even human, seawater, fresh water, hydrothermal vein, hot spring, even in the atmosphere, which is about 10 kilometers up to sea level. You can find bacteria. Of course, fungus may not be present in all of them. I think fungi, yes, but protista may not. What is hydrothermal vein? Hydrothermal vein. Have you heard about that in National Geographic? This is something that people discovered that, many 50 years ago, but they really study about hydrothermal vein is from the past 30 years, and they discover a lot of microorganisms living at the hydrothermal vein. What is hydrothermal vein? Basically, they're located at the seabed, the bottom of the sea, and there is a crack, meaning there's a hole that allow anything that underneath the ground to come through the hole and in contact with the seawater. So whatever underneath the ground on the seabed is something must be very hot, like lava, the mud, very, very hot, high temperature. So whatever the temperature underground, it can up to the fluid that can come out from here, it could be up to 400 degrees Celsius. So it's like boiling temperature, more than the boiling temperature, it's very hot. So when it come out, it's like a small volcano. So when you're actually in contact, the seawater is extremely cold. So when it is in contact with the seawater, suddenly it's very cold. It bring in, because when it come out, it bring in a lot of mineral. So the mineral will precipitate immediately when they contact the coal, and they precipitate as like this. So the mineral forming a chimney, like a chimney structure. So in this chimney, and of course when they are blowing out, you have the black smoke. The black smoke normally indicates this is a sulfide, right? Now, this is a small ecosystem, which means a lot for the researcher and scientists, because a lot of animals and the microorganisms living around these regions. And they are in a symbiotic relationship. You will find shrimp, crab, tube worm, even pompey worm around here, but no fishes around here. They are staying in this ecosystem. And the microorganism living either in the animals or the sea animal to provide the nutrients, and the animal will actually converting whatever the sulfide, this is black, the sulfide into something to provide to the microorganism. So those microorganisms, basically, they are chemotrophic. They're using the chemical to produce energy. Why people are interested in that? This is something that, which is not, they think that this can be dated, this kind of environment, billion years ago when we start to have the earth. So this could be the, people say, could be the origin of life. The origin of life, how we actually forming a cells with the membrane, is actually from the pore inside this chimney. So the very first microorganism, of course, everyone agree, right? The most simple and the origins of life could be coming from bacteria. And the bacteria is a single cells. And most likely are not any other bacteria, it's by a bacteria which utilize chemical to synthesize the energy. And this is what it is. All right, hot spring, of course, archaebacteria are staying in the hot spring a lot. Even some bacteria also staying in the hot spring. Atmosphere, yes, people found that, but 10 kilometers away from above us, yes, you still can find the bacteria. People have done that. They have used a filter to filter out the air, they collect the bacteria. But anything more than 30 kilometers above the sea level, there is no life of bacteria. And because of the less oxygens and also a very strong UV light, not possible, right? So they are staying everywhere around us. Okay then, that's all for today. And if you have any questions, if you want to take your attendance, please come to me.